Our next speaker, Dr. Michael J. Smith, is a professor of pediatrics and the interim division chief of pediatric infectious diseases at Duke University School of Medicine. During the pandemic, Dr. Smith has been an investigator for multiple vaccine clinical trials, including the Pfizer studies in both adults and children. He also serves as the Duke Principal Investigator for the CDC's Clinical Immunization Safety Assessment COVIDVax project. Dr. Smith completed medical school at the Columbia University College of Physicians and Surgeons and his pediatric residency at Hasbro Children's Hospital in Providence, Rhode Island. He completed fellowship training in pediatric infectious diseases at the Children's Hospital of Philadelphia, where he also earned a master's degree in clinical epidemiology. After fellowship, he served on the faculty of the University of Louisville before coming to Duke in 2017. Dr. Smith currently serves as the chair of the American Academy of Pediatrics section on epidemiology, public health, and evidence. Dr. Smith, over to you, sir.
Great, good morning, thank you. Just, I guess, to do the quick sound check. Can you hear me okay?
Yep.
And are you seeing, I always mess this up after, a year and a half on Zoom meetings. Are you seeing the full screen?
It says you've started screen sharing, but I don't see any slides yet.
Okay. I'll try that again. Yeah, it does say it's paused, doesn't it? Okay, let's try this again.
All right, I see slides. They're not full screen yet. There we go. Good to
go.
Okay,
great. Thank you so much for that introduction, Kate. And and thank you, Dr. Miller. Your passion is evident, and I think I just want to highlight that key point, which is, the communication that children and parents have with their child's provider is the single most important thing we can do to kind of boost vaccine confidence in the United States. Before I get into my talk, as Kate mentioned, I do have to acknowledge that I have been a site investigator here. Dr. Chip Walter is the PI for us here at Duke for the Pfizer vaccine trials. And I think while that's a disclosure that I have to announce, that does give me some a little bit of inside information that I can share with you about kind of how those trials were run and how we got here. And of course, I'm not going to intend any unapproved or investigative use. The vaccines we're going to talk are either fully approved for children or have an EUA down to 5. And I think, where I fit into this, I went to pediatric residency, thinking that I was going to be a general pediatrician. I was an intern shortly after that Wakefield paper came out that Dr. Miller shared. And this was a hot question, MMR and autism. And I quickly realized that I did not know enough about vaccine safety or the vaccine licensure, like how that all worked, to have these discussions with parents. And I did an ID elective and loved it. And so I am not the primary care provider that I thought I would grow up into becoming. However, I have been able to be involved in vaccine clinical trials and involved in vaccine safety at the national level. And so I think my role is a little bit different, which is to how do we create those data? So I heard Dr. Miller say, vaccines are safe, side effects are minimal. How do we know that? And those are some of the things that we'll talk about in the next hour or so. So when we're done, you should be able to describe the development and testing process for vaccines in the United States. You should be able to describe common adverse events following COVID vaccination. And you should be able to explain the post-licensure vaccine safety mechanisms in place in the United States. And again, I have a master's degree in epidemiology, and I start every vaccine talk, whether it's vaccine safety or vaccine clinical trials, with these slides. So remember the levels of evidence. It's a pyramid, and at the lowest level of evidence is kind of case reports, or it's almost like what you would read on the news. Someone did something and this happened to them. Okay, a little bit stronger is the case series, which is here's a group of people who had something happen to them, and maybe there is a causal association there. Now, these case series can be interesting, but they don't really prove anything. They may be useful for generating hypotheses. And when it comes to vaccine safety, sometimes we're right. So here was a case series that was published in May, shortly after we started vaccinating kids. And this was symptomatic acute myocarditis in 7 adolescents after the Pfizer-BioNTech COVID-19 vaccination. And it turns out that association was right. And then, of course, here's the paper that you already saw. I shouldn't even call this a paper. It's been retracted. Dr. Miller pointed out that this was a fraudulent paper, and that is totally true. The other thing I would say as an epidemiologist, even if it weren't fraudulent, which it was, we're talking about 12 kids. You should never conclude anything based on a story of 12 children. The next level of evidence are kind of observational studies, and these can come in two flavors. What we call a case control trial. It's when you start with an outcome of interest and you compare exposures between those with the outcome and those without, or a cohort study where you follow a group of individuals over time and you look at outcomes between those who had the exposure and those who didn't. So that's very common in vaccine safety studies. And of course, the highest level of evidence comes from randomized controlled trials. And these are similar to cohort studies, except the exposure of interest is prescribed, right? You get randomized to, for the purpose of this talk, a vaccine or a placebo. Randomization accounts for known and unknown confounders. It's considered the highest level of evidence and clearly is needed for licensure of vaccines and really all other pharmaceutical agents. But before you get to the phase 3 trials, you obviously need to do some work to get there. And this is a paper that Chip Walter, who I mentioned, is the CMO here of the Duke Human Vaccine Institute. He was the PI for the vaccine trials. He and one of my other colleagues published earlier this summer, but it's a nice summary of the steps. So there's some preclinical work. You need to come up with a concept and preclinical studies. So that's developing a rationale based on disease. You need to identify an immunogen, develop a manufacturing process, and do your preclinical studies, which includes acute toxicity, typically starting in animals first. And COVID was no different. I will say, just like Dr. Miller, I struggled. I'm going to talk about vaccine development in general or kind of COVID specifically. And we're going to do both. We're going to talk about vaccine development and using what's happened with COVID as an example. So you have to do these acute toxicity studies in animals. You include doses that are much higher than those used in humans. And again, this is true really for any pharmaceutical agent. Then you have to do in vivo immunogenicity testing in animals, ideally in juvenile, for us in pediatrics, rats, if you're going to do a pediatric study, or possibly primates. And there's certainly primate models for COVID that happen throughout the vaccine process. Then the company files an investigational new drug application before you can roll out the trials in humans. And over here, you can see kind of halfway up here, the clinical studies, typically divided as phase 1, phase 2, and phase 3 trials. And just to remind everybody what those mean, phase 1 are the early on trials. And again, I'm a pediatrician, so all of our vaccines usually start off in adults to make sure the vaccine's safe. Healthy adults, make sure the vaccine's safe, make sure the vaccine's tolerable, make sure you're aware of what the vaccine side effects are in relationship to doses, and does the vaccine lead to the desired immune response? Efficacy is not really something that you're focused on in a phase 1 trial. You then move on to a phase 2 trial, which has several hundred from a target population, if you have one. And again, these help you hone a little bit closely on what's the desired vaccine dose and the dosing interval. Is the desired immune response to the vaccine achieved? And you can start talking about some of these more common short-term vaccine side effects. And again, if you have promising phase 2 results, you move on to phase 3, and that's really where I'm going to spend the bulk of today's talk, going over some of the phase 3 vaccine trials, mostly in children, because I'm a pediatrician, but we'll talk about the adult studies as well. So the questions you answer here are, how effective and safe is the vaccine when comparing people who receive the vaccine to those who do not? What are the common side effects? Is the safety and immune response consistent across different lots of the vaccine? And although interestingly, we do not really have these studies yet for COVID, and we'll talk about that in a little bit, but many phase 3 studies, especially in younger children, integrate the vaccine of interest into the existing childhood immunization schedule. And you can answer these questions. What is the effect on safety and immune response when the vaccine is co-administered with other vaccines? Again, probably more of an issue for us on the pediatric side, although for COVID, there are some studies going on now about co-administration with HPV, HPV and COVID vaccine in teenagers, and in adults, influenza vaccine and COVID vaccine. So you do the studies, and of course, make that sound really easy. We'll talk about this more, as the hour progresses. But after this vaccine development and testing, there are several other layers of reviews and committees that the data need to be reviewed. So the first thing is that the company would submit to the FDA for a BLA or biologics license application. And remember, that's really only happened for COVID vaccine, the Pfizer COVID vaccine in people 16 and up, or in the case of a pandemic, emergency use authorization. And again, pre-COVID, this was kind of happening behind the scenes. You know, even as Dr. Miller mentioned, you know, we had a prior pandemic, which was the H1N1 pandemic. Social media was not as big there. This was not covered by the media, but it seemed like whenever these things came out with each COVID vaccine, the media was covering, you know, the VRBPAC. People didn't hear the VRBPAC until 2020 and COVID. But that's the Vaccine and Related Biologic Products Advisory Committee, which advises the FDA on the decision to license or grant an EUA. And I think the best way to describe what happens after an FDA recommendation is then it becomes possible or allowed to use the vaccine product in humans in the United States. Then the second question, though, kind of from a public health angle, and we talked about the immunization schedules in Dr. Miller's talk, you know, how do we create those schedules? And that's really where the ACIP comes in, the Advisory Committee on Immunization Practices, which is an advisory group that really makes a recommendation to the CDC about how should this vaccine be integrated into the existing vaccine infrastructure, and again, probably more so in pediatrics, but into the existing schedule. The CDC director then ultimately has the final decision based on the recommendations of the ACIP. And you know, usually there aren't any major discrepancies. I think the last one, perhaps when the CDC recommended boosters, there was some disagreement between the FDA and the ACIP and Dr. Walensky about who should and should not get a booster. So there, just to remind everybody, initially there wasn't agreement that everyone should be eligible for a booster. That became a little bit stronger after Dr. Walensky reviewed the data and made her statement. Of course, now with Omicron, really everyone is eligible for a booster at this point. So that's really kind of the FDA and public health angle here. And then, you know, for those of us who have our medical societies, again, the American Academy of Pediatrics also makes recommendations for their members. That comes from the COID, which is the Committee on Infectious Diseases, which it makes advisement to the AAP, who publishes recommendations in pediatrics. And similar things happen, again, for other professional groups, the AAFP for family practitioners, and ACOG for the OB-GYN, which is the OB-GYN professional group. And those recommendations are published in respective journals. Now, this usually takes a long time, years, at least 5 years, 5 to 10 years, people will say. And you know, I think one thing that comes up, going back to this question of vaccine hesitancy that you heard about in the previous hour, is this was done really quickly. You know, when you call something Operation Warp Speed, it doesn't necessarily invoke confidence in everybody. But this is one proposal. It's a nice opinion piece in New England Journal of Medicine that explains kind of how you can do things differently. And one thing, probably the big thing here, and I'll show you what happened with Pfizer here in a second. But you know, well, historically, when you're not in a pandemic and you have time to produce a vaccine, you know, while your phase 1 and phase 2 trials are going up, you're really just doing small scale clinical trial material. As you enter your phase 3, you're starting to scale up your manufacturing. And really, it's not until licensure that you do your large scale manufacturing. In the face of the pandemic, you know, these companies and the governments and the private entities that funded them really had to hedge their bets a little bit. So manufacturing was being developed and scaled up, you know, before, in some cases, you had the results of the efficacy trials. And large scale manufacturing was happening as the efficacy trials were going, certainly before they had the regulatory pathway for emergency authorization. The other thing that is important to know, you know, we talk a little bit about the mRNA vaccines are really the main vaccines. Some people, you may have seen this, call this kind of a plug and play mechanism. yes, while these are the first mRNA vaccines that have been used in routine clinical care, there have been lots of other mRNA vaccines in trials. So the technology has existed for some time. For instance, here at Duke, we were involved in a paraflu vaccine trial that Moderna is still leading. Obviously, it got bumped for COVID, but the technology exists, and all you really need to do is use different mRNA. In this case, for the spike protein, and you can incorporate that into existing vaccine technology. So that's another reason that this was able to happen so much faster. And of course, the other thing, as I mentioned, I have a degree in epidemiology. On my very first day of class, one person said, if you want to design a successful trial, you need bad outcomes. So as a clinician, we hate that. As public health people, we hate that. But as a trialist, bad outcomes drive your studies. And when you do a study in the middle of a COVID pandemic, you have lots of disease. It becomes much easier and much faster to show that a vaccine works. And again, a couple other things here. This is from Nature, just a snapshot of kind of how Pfizer did this in a year. I think I talked about a lot of these points already, but the other thing that Pfizer did was you can roll your phase 1 and 2 trials together and your phase 2 and 3 trials together. And at Duke, we were fortunate to actually enroll the first pediatric patients into the Pfizer trials, and those started off as really phase 1, phase 2 dose finding studies. And the protocol was written with kind of 2 phases. That it was written ahead of time. Here are the doses. Then there's going to be an interim analysis. We'll pick which one works, and then we're going to move right into phase 3. So that helped save quite a bit of time. And it also helped us identify patients really quickly. Again, here in the Triangle of North Carolina, we're not a particularly vaccine hesitant area of the country or the state, for instance. And we had no trouble enrolling people into this trial. I've done a lot of vaccine trials in my career, and there was a wait list, in the thousands. So when you have lots of people and a protocol ready to go, you really can make things more efficient without skipping any of those steps that we just went over. And I think you've probably seen this. Is probably the 3rd or 4th talk where you've seen this. Just remember, for us in pediatrics, it's really just the Pfizer vaccine that we have an EUA down to 5. Moderna greater than 18, and then the vector vaccine, which is the Johnson & Johnson vaccine. I'm not going to talk about too much unless you all have specific questions at the end, but that is approved and used for adults only. So again, I see my role as an academic infectious disease doctor to help create these data, help find the data on vaccine efficacy, safety, and immunogenicity, which really are, and we'll talk about Pfizer as an example, which really are the most important parts of these trials. So efficacy, what is the incidence of infection in vaccinated versus unvaccinated individuals? Immunogenicity, what is the immune response in vaccinated versus unvaccinated individuals? And then safety, what are the short and long-term adverse events in vaccinated versus unvaccinated individuals? And those are really the 3 questions that most vaccine trials try to answer. Again, efficacy can be hard if you have a disease like meningococcal disease, which only occurs in 1 in 100,000 people. But if you have, a pandemic or a new vaccine, like the rotavirus vaccines, then efficacy usually is the primary outcome. So I want to walk people through the Pfizer trial, and just so everyone's aware, Pfizer started in adults, then they went down to 16 using the same protocol, the 30 microgram dose, and then they went down to 12. So it was essentially the same protocol for the 12 and up crowd. Different protocol for the 5 to 11, as I mentioned earlier. But you have to be a healthy individual, and that's important because we don't really have randomized controlled trial data on people with a lot of chronic illnesses. The other thing, as I mentioned before, when you're designing a clinical trial, you really want to find people who are going to have your outcome of interest. Earlier on in the Pfizer trials, at least, they kind of relaxed this once the age went down. You had to be at a higher risk of contracting COVID-19 because they wanted to meet their efficacy endpoint sooner. So some interesting exclusion criteria to keep in mind as we talk about vaccine hesitancy and figure out how to vaccinate the population is that if you had previous clinical or microbiological evidence or diagnosis of COVID-19, you are not allowed to be in the trials, again, at least for 12 and up. If you were immunocompromised with known or suspected immunodeficiency, either due to an underlying diagnosis or to a medication that you were taking, you could not be enrolled in the trials. And if you were pregnant, you could not be enrolled in the trials. This is important to consider as we talk about later, kind of post-licensure or post-EUA surveillance, making sure we really understand how well these vaccines work in these populations who are excluded from the trials, which are really important. This is just something I wanted to share too, probably less relevant to the overall discussion, but a temporary delay criteria. One is if you had something like COVID, you cannot enroll in the trial. And again, this is another key thing I want to make sure everyone's aware of, that if you receive seasonal or pandemic influenza vaccine within 14 days of your enrollment or your potential enrollment, or any other non-study vaccine within 28 days before study intervention administration, that was an exclusion criteria. That's interesting and important because, of course, the CDC recommendation and the ACIP recommendation is that you can give this vaccine concomitantly with other vaccines. We're gathering data on that, as I mentioned before, but not from the randomized trials. And then, of course, if you received systemic corticosteroids less than 14 days, that was another contraindication. So again, if you are actively getting an immune modulator, you cannot be in the trial, which makes sense when you're trying to find immunogenicity and efficacy, but leaves us with some questions as we try to figure out the best way to vaccinate these folks. There's a lot on this slide, but this is, again, really now getting up under the hood of the Pfizer vaccine trial and kind of what happens to a study participant. This is a question that people ask me. Again, I'm not in private practice, so usually the people that come to my office or call me are kind of extremely vaccine hesitant and have lots of very specific questions about the nuts and bolts of these trials. And this is some of the stuff I share with them. So you come in on day one, you get all this screening that I just talked about done. And probably two important things happen. One is you get your baseline antibodies. That's to see if you have evidence of COVID vaccine. These are not available in real time. So again, you could have had a previous silent infection or asymptomatic infection in the trials and be still included in the study, but not a known clinical diagnosis. And you also get a swab because you want to see if people, have asymptomatic COVID during the trial, to help you better understand whether it's the vaccine that worked or their infection that caused the problem. Then you come back about 3 weeks later for a second dose. Notice that you get another PCR there, but no blood draw. It's not till a month after your visit 2 that you come back for that blood draw. And as we talk about the emergency use authorization and immunogenicity, it's really how immunogenic is 2 doses of these vaccines. That's what we know. We don't really have immunogenicity data after 1 dose. We have some efficacy data. And how do we have efficacy data? Because if you have a sick visit, you have a list of symptoms, cough, runny nose, things that seem like an upper respiratory infection, you need to come back for a study visit, kind of early on when they started these trials, back in the summer of 2020. And then those eventually became at-home self-swabs with telemedicine. And understanding how many sick visits there were and how many sick visits were COVID in the vaccinated as compared to the unvaccinated arms. That really is what drove what we know about efficacy. And those nice curves, I didn't put them in the chart, but the nice curves that you may have seen in the New England Journal papers for Pfizer and Moderna. Now, so that's kind of the overall study. But after day 1, other things happen. So again, how do we know what the side effects are after these vaccines? And this is where we come and talk about adverse events. So from an FDA perspective, what is an adverse event? It's any undesirable experience associated with the use of a medical product in a patient. Then it gets graded for severity or intensity and assessed for relatedness to the study product. So again, these are the things that you probably are all aware of. But you look at 2 things. You look at local injection site reactions, so things like pain, tenderness, swelling or induration, redness. Those get graded for intensity. And either the participants themselves do this if you're an adult, or the legally authorized representatives, that's what an LAR is, provides these data electronically or during an in-person visit. This is an example of kind of what we collect for some of the studies we do. This is the paper version of the diary. And again, kind of each day you come and get your vaccine, and that night you check all these things. You get a ruler and you measure how much swelling there is, how much induration there is, and you check these mild, moderate, severe, or grade 4. Most studies usually have 3 grades. So mild is something that's noticeable but does not interfere with activity. A moderate means it interferes with activity but did not need a medical visit or absenteeism. And then 3 is kind of severe, prevents daily activity and or resulted in a medical visit and or absenteeism. And then 4 is, emergency room visit or hospitalization. I've seen this both ways, but again, when you see these 3 grades, this is what I'm talking about. Systemic reactions similarly are assessed for a fixed period of time. Most commonly the first 7 days after your vaccine. We ask people to take their temperature. If you're an adult, kind of ask these symptoms: fatigue, body aches, joint aches, headache, nausea, vomiting, diarrhea. A little bit harder for those of us who do pediatric trials. So you'd be a little bit more vague, perhaps, irritability, although vomiting, diarrhea, I guess, is pretty objective. But, lethargy rather than body aches or joint aches, decreased appetite, and irritability. And again, this can be captured, electronically or on paper at a clinic visit. So again, same thing. This is the bottom part of the paper diary. We also do this electronically. So this is something you can just do on your phone. And really, I'd say 99% of our study participants for the Pfizer vaccine and most of our clinical trials now prefer to do this either online or on their phone. So there's a screenshot of the iPhone, and you can kind of see it's very easy just to kind of click these radio buttons. And in most studies, if there's anything on here that's concerning, if there's any grade 3s or other symptoms to report, that does trigger a phone call. And that's how you kind of keep abreast of what the vaccine adverse events are. And each trial has their own adverse events that they're interested in. So those are called kind of the solicited adverse events. We want you to look for these 3 things, and we're asking you specifically about them. You also have unsolicited adverse events. That's this question here. Any other symptoms to report? Did you go see the doctor for anything? You grab that information. Did you have an adverse event of special interest? And so these 2 usually are longer than the 7 days. So adverse events of special interest related to COVID vaccine, for instance, typically tend to be, myocarditis, MISC, things like that. And you're going to study that for the duration of the study, which is often, 6 months, 12 months, 24 months. And again, probably 24 months for the major phase 3 trials. And did you have an onset of a new chronic medical condition? So that's what we talk about when we talk about adverse events and how you find them. The other thing that you'll see in these papers and in the FDA briefing is a serious adverse event. And again, that really is an FDA kind of regulatory term. So when you see SAE or serious adverse event, that is an adverse event that results in death, is life-threatening, results in hospitalization, disability or permanent damage, a congenital abnormality or a birth defect. These are things that you can kind of see here. So that's what's meant by an SAE. It has nothing to do really with necessarily the severity, unless you end up dying or being admitted to the hospital. And then you have to grade or assess causality. And some protocols have specific criteria for how you do this. Often it comes down to the principal investigator kind of discretion and experience, but here are some things you think about. Did it occur during a plausible time following vaccination? Does it correspond to those previously associated with a particular vaccine? Is there a strong biological plausibility for this being an AE? Do you have a lab result that shows it's the case? Did it happen again after re-administering the vaccine? So these are all little things that you'd want to think about as again, initially the principal investigator, but also the monitors and eventually the study team will consider when you're trying to figure out whether an adverse event is in fact causally associated with the vaccine or not. So going back to the Pfizer trial, I talked about the safety here. Efficacy, what they chose was efficacy against confirmed COVID-19 occurring from 7 days after the second dose. So kind of when you're not fully vaccinated in terms of the CDC definition, which is 14 days, but at least a good week after dose 2. And then, for immunogenicity, immunogenicity for the younger children, at least the 12 to 15-year-olds and later the 5 to 11-year-olds, was compared to participants 16 to 25 years of age. And that's kind of what we call immunobridging. So you know the vaccine is efficacious in that age group, and you get the can you generate the same immune response in 12 to 15-year-olds as you did in 16 to 25-year-olds who had efficacy, and then in 5 to 11-year-olds. And I don't think I say this anywhere else, but remember the 5 to 11-year-olds got a lower dose, which is 10 micrograms, 10 micrograms, and had similar immunogenicity, which is just awesome. And again, Dr. Miller mentioned the efficacy outcomes were 100%. I fully believe this is an efficacious vaccine. Just remember, you have to be a little bit careful because there weren't many cases of infection. You know, you're talking about the in people with no previous infection, 16 in placebo as compared to 0 who got vaccine, and there are pretty wide confidence intervals. So yes, it's 100% on paper, but probably a little bit less than that. That just happens because by the time these trials were done, COVID was kind of at a dip, which is why relying on the immunogenicity and immunobridging for the EUA is so important. So I already talked about this, and in both groups met non-inferiority objectives. So again, just to show the results of local reactogenicity, and again, I'm a pediatrician, so I'm going to focus on the pediatric patients. You know, it hurts. It hurts a little bit more the younger you are, and I think we've seen that anecdotally, right? I'm a professor of pediatrics in my mid-40s. I work at an academic institution where I have junior faculty, fellows, residents, and medical students. If you were a medical student, resident, or fellow, you were more likely to actually miss work from this vaccine because you had a fever and you couldn't come to work, you know, with a fever. That I kind of show here in the 12. Sorry, this is the 5 to 11-year-old group, and again, much better tolerated in the 5 to 11-year-olds. When you look at systemic events, I think it was all, as we've all seen, dose 2 is a little bit worse, particularly, again, this is 12 to 15-year-olds and 16 to 25-year-olds, particularly for, you know, headache, a little bit worse, the chills were a little bit worse, and then these muscle pains, so these flu-like symptoms, a little bit worse after dose 2. Interestingly, when you look at the pediatric studies, so this is the 5 to 11, after dose 1, you hardly see anything. In fact, for fatigue and headache, you don't see any statistically significant difference between the kids who got the real vaccine and placebo. Again, on paper, more people with the placebo got the headache. And I will say, going back to this, the importance of trust. I'm a father, and my daughter got her vaccine. She's 11 years old. This was our experience. Had she been in the study, I would have thought she got the placebo. Dose 2, a little bit worse, but again, not nearly as many side effects as you see in the 12 to 15-year-olds who got the 30 microgram dose. And again, we were part of those phase 1-2 studies in the 5 to 11-year-old kiddos here at Duke. And I will say, the young children who got the 30 microgram dose had a heck of a time of it. And that's how we got to the 10 microgram dose for that age group. I do want to talk about Moderna vaccine real quickly, because this trial has been published. So, Moderna has submitted to the FDA for an EUA over the summer. It's not yet been granted, but this study is out there. And I think also just kind of as a second example of how similar all these trials are. Moderna took a slightly different approach. However, their main trial was 18 and up. So they have a 12 to 18-year-old trial that I'm going to talk about very briefly here. And then they also have trials going down to 6 months, just as Pfizer does, that are ongoing. Very similar criteria. You can't be included if you were pregnant, if you had prior COVID vaccine or current treatment, if you were immunocompromised. So again, a lot of the same missing, I wouldn't say missing data, but kind of gaps in knowledge here. Interestingly, for the Moderna trial, if you travel outside of the US within 28 days prior to screening, that was also exclusionary. And same things about if you had an illness or if you had received another licensed vaccine within 28 days. So again, basically you've seen one protocol, you've seen them all. This was a very similar study, except instead of 21 days between dose 1 and 22, you have 28 days. And again, what I want to show here is, you have the review of the AEs. You get a blood sample for vaccine immunogenicity pre-dose 1, but not again until day 57 or a month after, after dose 2. So the immunogenicity is based on 2 doses. However, they did get a swab at visit 1 and visit 2, and if you got sick. Again, just to show here, so the Moderna does have more antigen, so it is a little bit more reactogenic. Again, these are the 12 to 15-year-old data, so you can see mostly pain at the injection site, and then just as we saw for Pfizer, particularly after dose 2, 75% of people had headache, fatigue, and chills. And this is really all we've seen in these trials. So again, to bring home Dr. Miller's point, I wanted to show everybody how do we get this side effect data? This is how we get it. So when you put these 2 randomized controlled trials together, or 3, I guess, since you're talking about the 5 to 11-year-olds as well, we know that COVID vaccines are efficacious, more so from the adult trials that show that 95% efficacy, those nice Kaplan-Meier curves that you remember. We know that COVID vaccines are immunogenic, and we know that there are really no major safety concerns. However, there are several things we don't know based on these randomized controlled trials. What's the impact of prior symptomatic infections? Now, in the younger group, you could be included, the 5 to 11-year-olds, but if you're a teenager or up, we don't know that. What is the impact of concomitant vaccine receipt? What about immunogenicity in immunocompromised individuals? Is it safe in pregnant women? Pregnancy was an exclusion criteria for the trials. I will say the answer to this question is yes, and we know that because even though you were not supposed to get pregnant during the trial as part of the consent procedure, you agreed not to do that. Certainly, there were a handful of women and men whose partners got pregnant, and those women are being followed in multiple registries from the trials. There's also been some post-surveillance trials that I'll tell you about in a little bit, looking at the impact of this vaccine both on pregnant women and their children. And what's the impact of race? Is race something we need to worry about? I don't personally think there's any differences in the safety of this vaccine by race or ethnicity for that matter, but just be aware that at least in the pediatric studies, these were not particularly diverse trials. The Pfizer 12 to 15-year-old study was 85% white participants, only 5% Black, 12% Hispanic. Very similar with Moderna 12 to 15. The Pfizer 5 to 11, similar kind of racial breakdown. There's some more people of Hispanic ancestry, but that's because they recruited from Spain. So, from a scientific perspective, I don't know how important this is, if there are differences, but when you consider the impact of COVID in our Black and African American populations and our Hispanic and Latinx populations, they certainly were not studied as enough as they should be. And I think post-surveillance studies can be very helpful, to make these populations and these groups of individuals feel that they are part of the scientific process here and the vaccines are safe. Okay, so in the last 20 minutes or so, I do want to talk about post-licensure vaccine safety. So we talk about kind of the steps to designing a trial and getting approved, or formally approved or EUA. But once a vaccine is approved for use in the population, it doesn't stop. You remember from my earlier slides, there's kind of what we call phase 4 trials, and those are usually run by the pharmaceutical companies as part of a condition of the EUA or the BLA. They have to do safety studies. But also, there are several safety mechanisms I'm going to tell you about. And it's important to keep doing vaccine safety surveillance to identify rare adverse events that may not have been found in trials that only excluded tens of thousands of people. And again, to find adverse events that may have occurred in individuals who are excluded from the trials, those groups that we just mentioned. And the good news is now there are almost 500 million doses of COVID vaccine administered in the United States. You can see here, mostly Pfizer, followed not too far behind by Moderna, and very fewer, but still 17 million people. It's a pretty good sized denominator have received the J&J vaccine. For us in pediatrics, those numbers aren't quite as big, but for 12 to 17-year-olds, and this is data from the AAP website from last week, we have 15 million 12 to 17-year-olds who have received at least one dose, about 13 million who are fully vaccinated, and that should help us find rare adverse events in these populations. We're getting there in kids. We're only a month or so from the ACIP approval and recommendation of the vaccine, but we have now just under 5 million children 5 to 11 who received their first dose, about 18% of kids in that age group. So for those of us who are are in the pediatric vaccine safety world, this is a critical time because these kids are now, you have about 5 million children who are in the process now of getting their second doses. If we're going to see things like myocarditis, if we're going to see things like MISC, it's probably going to be in the next couple of weeks. So I don't think that myocarditis is going to be common in this age group because we don't really see myocarditis in younger children. And also the dose is lower. It's a third of the dose. And we've already seen, based on the data I've shown you, that this vaccine is much less reactogenic. So causes very, very few side effects, yet is still immunogenic in this age group. So I think it still is a theoretical concern. And as more and more of these kids get their second doses and that number increases, I'll feel much better about that statement. So, and Dr. Miller touched on some of these, but there are several longstanding post-licensure mechanisms in place in the United States. There's VAERS, there's the VSD, which is the Vaccine Safety Data Link. There's CISA, which is the Clinical Immunization Safety Assessment. All of these have existed pre-COVID. VSafe, which is kind of the handheld app after vaccine health checker, that really came of age during the COVID pandemic and has provided some really nice vaccine safety data. So Dr. Miller talked about VAERS. I will not go into this in too much depth, but it's, basically anyone can report to VAERS. It's an early warning system. And, when you have 500 million people vaccinated, it's really the best way to find rare events. Now, remember, just to reiterate this point, although Dr. Miller did say it earlier, there's no comparison group. Only patients who received a vaccine and had an adverse event are included. And, going back to my early slide about hypothesis generation and levels of evidence, you can certainly use these data to generate hypotheses, but they do not prove causation, similar to every other kind of case series that we review in medicine. When you look at VAERS, you can kind of see some of the reports. These are, this is a survey, a collection of VAERS reports from adolescents. And, most of the reports were non-serious. But that's where you start to get a better sense of, of myocarditis. So again, I already showed you how many millions of teenagers have been vaccinated, but of those 863, there were 863 reports that included things like chest pain, increased troponin, and myocarditis, really with negative SARS-CoV-2 test result, which means they didn't have an active COVID infection causing this. And the hypothesis was that this was the vaccine. Again, this is not a perfect, rigorous epi-analysis because it's just doing the best you can do with VAERS, which is based on the number of doses administered. What's the general risk factor or stratification by age and sex? I just want to point out here that this is much less common in girls or teenage females as compared to males, and it's most common in the 12 to 29-year-old group. So VAERS strength, though, it's nationally representative, it's rapid, and again, I think we've touched on these points, so I can kind of speed up a little bit here. But again, there's reporting bias. Things may be underreported if you don't think about the vaccine, and certainly can be simulated reporting. And just one thing, going back to the previous discussion about vaccine hesitancy, there was a really interesting paper now about 10 years ago where it was found that people who put in autism as a VAERS report, it wasn't parents, it wasn't physicians, it was actually personal injury lawyers. So the downside of having anyone be able to report to VAERS is that there's going to be some bias. And again, there is no unvaccinated comparison group. So VSafe, again, it's a nice way for kind of real-time updates. But again, only vaccinated people. But you can again see how many kids you have. All of a sudden, you now have information rapidly collected on 50 to 60,000 some kids. And we won't read all these, but for the most part, these line up very nicely with what we saw in the vaccine clinical trials. I want to talk a little bit about the Vaccine Safety Data Link. And as an epidemiologist, going back to the beginning of the talk, VSD really allows you to do a cohort study. It's a cohort. What is the VSD? It's a partnership between the CDC and several geographically diverse managed care organizations. Includes clinical data, including vaccine records for nearly 10 million individuals each year. And VSD data are updated weekly. They allow for a better understanding of causation because you can compare rates of an adverse event of concern in people who are vaccinated as compared to those who weren't. So there really is a control group here. Here are the sites. So Kaiser Permanente, really all over the West Coast and in Colorado, Health Partners in Minnesota, the Marshfield Clinics, and Harvard Pilgrim work together with the CDC. And I think that the best way to kind of explain VSD is to show you this paper that was published in JAMA in September. This is by Nikki Klein, who's at Kaiser in Northern California. And it's called Surveillance for Adverse Events After COVID-19 mRNA Vaccination. And this is really the preliminary analysis of what's ongoing work and how this was stood up to assess vaccine safety. During the study period, which was from in 2021, since the vaccines were approved through about the end of August, there were 12 million doses of mRNA vaccine administered to 6 million individuals, and pretty close to half-half split, almost 7 million Pfizer, 5 million Moderna. The population at the time of the study was 12.5 million people, so just under 4% of the US population. And they use multiple comparators. So I mentioned the strength of theirs. You can actually look at things that happen in vaccinated as compared to unvaccinated people, but because that's not a randomized controlled trial like the vaccine trials are, there could be some confounding there. Might people who choose to be vaccinated be different than those who choose to not be vaccinated? And I think Dr. Miller showed us, yes, there are differences by race and political affiliation, for instance, and lots of other medical things. So actually for this trial, the main outcome was the incidence rates in days of 121 after people were vaccinated, as compared to days 22 to 42 in vaccinated comparators. As the study goes on, some of the outcomes can be adjusted to look in unvaccinated and vaccinated as well. Just to show you, again, these were all pre-specified. If you go back to the CDC meetings from December 2020, these were all on there. These are many of the things that they look for. ADEM, MI, appendicitis, Bell palsy, which has been seen in other vaccines, venous sinus thrombosis, seizures, DIC, Guillain-Barré, GBS is something that Dr. Miller had mentioned, has been seen in other vaccines. ITP, certainly seen commonly in MMR, not really in any other vaccines, but all these things they're looked for. And, and when you have 13 million people, 10 million of whom have been vaccinated, you really have power to look at some of these things. And the good news is, despite having that power, none of these adverse events met signal criteria. They also did kind of descriptive monitoring only for some other things like anaphylaxis and MIS-C. And I will point out that anaphylaxis was seen. There are 183 reported cases. This is based on billing codes, ICD, well, at the time of the study, ICD-10 codes. And because everyone's, as part of an MCO, you can actually get access to those medical records and review them. So 55 of those were confirmed. And just what do we know about anaphylaxis in vaccines? They were almost all in women, which is very typical and common for other vaccines. It was also seen earlier on in the pandemic. Median of 10 minutes after vaccination. And that translates to about, 5 cases per million doses. And that order of kind of, 1 to 2 to 3 episodes of anaphylaxis for most routine vaccines, is pretty similar to what we've seen in the COVID vaccine so far. Understanding that this data set continues to grow. And really, these data are reviewed on a regular basis. Just to talk about myocarditis briefly, there was no signal seen in the overall cohort. They did a post hoc review of members 12 to 19 and found 34 cases of confirmed myocarditis. I think we've heard a lot about myocarditis. You can certainly talk about this in the Q&A session. But again, median age is 24 years, 13 to 29, almost all male, 85%. Most were hospitalized, but median length of stay for 1 day. And again, with myocarditis, most individuals do really, really well. And here's the time frame. If you're gonna get myocarditis, much more common in the first 7 days after the vaccine. Again, which is why as more and more younger children get vaccinated, as each day goes by, I'm feeling a little bit better and have a sigh of relief that hopefully we will not see this in the younger children. I wanna finish talking about CISA, which is the Clinical Immunization Safety Assessment. And this is a long-standing partnership between CDC and 7 medical research centers that provides expert consultation and conducts clinical research on vaccine-associated health risks. I've been talking about vaccine hesitancy and vaccine risk communication for years, and I've written about CISA I didn't really understand what it was until I came to Duke a few years ago, and now have been able to participate in this program. So really, it's twofold. One is it's an expert consultation service. And if you have someone who has an unusual complication after COVID vaccine or any vaccine, go to the CDC website. I'll put it in the chat later. But if you go to CISA, you can request a consult, and it'll be reviewed by CISA staff and then brought to groups of not only infectious disease doctors and vaccinologists, but OB-GYN experts. We have lots of allergists now, anaphylaxis. We have a whole panel of hematologists with TTS. It's really an expert review, almost like a grand rounds review, in-depth review of specific patients. CISA also does post-licensure studies. So here at Duke, we're doing a maternal and pediatric cohorts. Again, we're collecting prospective data on children who are vaccinated, looking at including children who had past infection, because we don't know that. We're including people who are non-white and Hispanic, because we don't have as much data as we'd like in those populations. And CISA is also doing a randomized controlled trial looking at concomitant versus serial vaccine. And that's really flu plus COVID at the same time or given separately. What's the impact on immunogenicity as well as side effects? So in closing, I hope you realize that the vaccine development process includes multiple stages and review processes. And while these were modified to maximize efficiency during the COVID vaccine pandemic, really no corners were cut. All the pieces were in place and all the trials were done as they should have been. I think based on the published clinical trials, I'm comfortable saying COVID-19 vaccines are efficacious, safe, and immunogenic. However, post-licensure or post-EUA safety surveillance has identified some rare adverse events after COVID vaccine, notably anaphylaxis. There's a big safety signal. Again, VAERS picked this up. We figured out what the contraindications should be, and rates of anaphylaxis have decreased. Myocarditis also picked up in VAERS, and then of course TTS after the J&J vaccines. So I'll stop there. I have my email for any questions. And I'll also be at the noon panel. Thank you so much for your time.
Thank you, Dr. Smith. So first question I have is, of course, will your slides be available for?
Yeah, absolutely.
Fantastic. I know that question is going to be asked, so just get that out of the way. So first question, do you think the pandemic has changed the process of vaccine development and manufacturing, or will we go back to the pre-pandemic process in the future?
Yeah, that's a great question. I think the technology is here, and again, it's not my term, but we kind of read a lot about kind of the this plug and play approach, at least for mRNA vaccines. Both Pfizer and Moderna are looking at how well their vaccines work against Omicron or whatever the next variant's going to be. And the really cool thing about this technology is if you have enough mutations, so Omicron has like 30 mutations to in the spike protein, you can actually use the same exact technology and build an Omicron vaccine. So I think in the context of a pandemic, whether it's COVID or the next pandemic, we've learned how to ramp things up and really respond really more quickly than we have with other vaccines. So in the face of a pandemic where it's really important to protect people, you can do that. I think if you have a disease that's not a pandemic, that's more indolent, you probably could take, and things got pushed aside. I will say, we're doing lots of other vaccine clinical trials that those studies have slowed down because there's been so much emphasis on the FDA and the ACIP on COVID that we haven't had time for some of these other things. I think the risks, going back to vaccine risk communication, the risks and benefits change a little bit in the face of a pandemic that's killed as many Americans as COVID has. So I think it's feasible. I don't think this is going to be the norm for everything, but I think what we've learned here will help us with future pandemics.
Thank you. Question from the audience. Why would the FDA ask for 55 years to delay the release of data and information used for the approval of the Pfizer biotech vaccine as per the filing in U.S. District Court?
I do not know about that, but I'd be happy to kind of read about that, and maybe we can bring that up in the noon discussion. Again, I'm not sure. I'm sorry, I'm not familiar with that one.
Fair enough. We will table that one. And then what's your feeling regarding journals and dialogue, questioning, or expressing concerns about vaccines being censored?
So I think journals probably shouldn't be doing that from an ethical perspective. I think the other thing that we learned in the pandemic, as long as we understand what we're looking at, we have preprints now, we have press releases. I think a lot of what we learned about these vaccines initially came in press releases primarily from the vaccine manufacturers themselves. I think you have to take those with a grain of salt. I think social media, there's a lot of, censoring. There's some issues, around that. And I think, should true misinformation be censored? I kind of leave that to those companies. But I think, at least within academic medicine, certain things like, what are the risks and benefits of vaccines for certain age groups? That's something that, several people have asked, and they're different. And what's interesting is, for me, at least here in the triangle, where we have lots of people who are excited about this vaccine, it went from this is too fast, this is too fast, this is too fast, to okay, when are we going to have this for kids, under 5? So the perspectives have changed a little bit. So I'm sorry that those are both kind of two hard questions that I have to come back to.
That's fair. You're allowed. It's putting you on the spot. Were there any prior mRNA vaccines that made it to late human trials? And if so, what did they learn?
Yeah, so I think my personal experience was there are several that Moderna has been doing. And remember, Moderna is a play on kind of mRNA in there. That's their whole platform. So they've existed for, a decade or so, doing this. In terms of late vaccine trials, so some of them, so the one that I'm most familiar with, because we were doing it here at Duke, was parainfluenza and a human metapneumovirus. Those went through phases 1 and 2. They were in phase 3. No major side effects seen. But again, since they didn't get out into practice, we don't have the hundreds of millions of people who received the vaccine to kind of know about more rare side effects. They were immunogenic and safe, using the parameters of safety that I just kind of showed you kind of within the 7 days after vaccine. And there are a couple other on their website that you can look. Moderna has been pretty transparent about their processes.
Great. Last question. Do you anticipate the COVID vaccine being given every year similar to the flu vaccine?
Yeah, this is a very common question, right? And the way it's going, probably. And we can boost people. What we're probably going to need to do is boost people, with a different vaccine each year, like influenza. Again, I don't have a crystal ball. I don't know that for sure, but with what we're seeing in this year, I would say it's likely that we're going to need an annual COVID vaccine. Now, people are working on kind of other things. It's just the spike proteins. And again, I don't think I mentioned this. Another reason that this was able to happen so quickly is, we knew from vaccine candidates from MERS, SARS-CoV-1, that the spike protein was a really good target antigen, based on prior work. So we kind of knew what we were doing. But there are other studies looking at different vaccine platforms that, just as we're looking for a universal influenza vaccine, it's not implausible that there'll be a universal coronavirus vaccine in the future. But I think in the next year or two, we're probably going to need, I wouldn't even call it a booster, like an Omicron or whatever the next, 5th letter is. I think that's probably likely.
All right. Well, thank you very much, Dr. Smith. We appreciate you coming up to Delaware from North Carolina for us.